And that's where I started to learn about the biology of a condition called Fanconi anemia. I was working on it in plants at the time. And then later, that's where I started to think about the genetics of this condition and the mechanisms of how it works in the context of humans as well as in plants, which is where I was working at the time. And I made a strategic choice to eventually come over to research in a medical field rather than in plant genetics. Hi, my name's Wayne Crismani and I'm a lab head here at St. Vincent's Institute of Medical Research. I run the DNA repair and recombination laboratory. I grew up in Adelaide and that's where I did all of my studies all the way up to my PhD. During my PhD, I was lucky enough to spend 4 months in France in a research laboratory. I had a great time there and I spoke to the person who was supervising me and said that if there was the possibility to come back for a postdoc, I'd really love that opportunity. And as luck would have it, I was able to go back and do a postdoc there, and that was a really formative time in my career. I had an excellent mentor called Raphael Mercier, and he's someone who really taught me about the power of genetics, doing it the right way, and it's where I really appreciated that genetics is a field of science where you can get really clear answers to very tough questions, in contrast maybe to some other parts of science which are a bit harder to sort of pin things down. And I was very lucky to have a number of good papers come out of that, in part due to Raphael and an excellent team that I worked with there. After that, I then left academia for a couple of years and I went to industry in the US and I worked for a large agrochemical company called Pioneer at the time. And this was another extension of my work into genetics and to see how things can really work at scale. It's quite different from medical research, but nonetheless, I took a lot away from it. After working for a while in the US in industry, I made a strategic decision to return home to Australia, but also to pivot and change fields slightly. I stayed working on the biology of this condition called Fanconi anemia, but I moved from plants to medical research. And while that really can be a jump in some respects, in other aspects it wasn't a big jump because the underlying fundamentals of genetics are the same whether we're talking about plants or humans. And so the knowledge that I developed about these processes in plants set me up very well for doing genetic research in humans. In my team, we work on Fanconi anemia, which is a rare genetic condition, and it can be quite hard to diagnose sometimes. Not only the diagnosis, but it can also be hard to find the genetic cause of the condition. And this is something that we're really interested by. It can be hard to diagnose for a number of reasons. One is because it can be hard to suspect the condition in the first place based on the clinical presentation. But then the follow-up to that is even after someone gets a positive diagnosis, it can be hard to find the genetic cause of the condition because there are so many genes that are involved in the underlying biology. And something that we have been doing for a number of years now is helping patients who have very tricky cases of Fanconi anemia to help them get a clear genetic diagnosis for their condition. So we're really focused on the edge cases. And the reason that I think that this is important is because understanding what makes these cases so tricky to diagnose teaches us something about the condition and also helps not only the family that is affected by this condition, but it will also help the next people who also have a diagnosis and a tricky case because we'll be better set up with the tools to improve the diagnostic pathways. At the moment, my lab is funded by the National Health and Medical Research Council. I also have a fellowship from the Victorian Cancer Agency, and we are lucky to receive some support from philanthropic sources and sometimes from smaller foundations. Over my career, I have been lucky enough to receive funding from other organisations such as Maria Revolt's Vision. I've had a large collaboration with Pfizer, an industry partner, and again, other smaller foundations have supported us and provided really important seed funding to get our projects to a point where they're competitive for NHMRC funding. I think it comes from two places. More basic passion comes from the fact that I really like genetics, and I know that if we approach things in a systematic way, we are going to find the answers to the problems that we're tackling. But something that has happened over more recent years is I've got to meet a lot of families who are affected by Fanconi anemia, and that is a massive driver and can really help us to dig that little bit deeper each day, to work harder and to think more strategically about what we're doing and how we're going to get answers that really help them. One thing that I have noticed with this interaction with the families and patients who are affected by Fanconi anemia is it really crystallizes your ability to make decisions about what you work on versus what you don't work on. And that's not something that I had appreciated in the earlier stages of my career. I think these days we have very little free time, so I'm really enthusiastic to spend as much time as I possibly can with my family. I have a very young family, so that's really what I want to do when I have 30 minutes or an hour free. But in terms of more hobbies, when I was growing up, I really loved playing music and guitar, and so that's something that I try to find a bit of time to do these days. And a more recent hobby that I have picked up is building guitar pedals, which has been something that I've really enjoyed. I've lost a lot of amplifiers in the process, but it's been a very cool learning experience, and I think that there's probably something there that's sort of linked to the logic that I really enjoy with genetics, how there's very clear yes-no answers. I think that building something like an electronic little guitar pedal, there's something quite similar and exciting about making something work, and you have that moment where you say, it's like, I've got it.